Okay, welcome everybody. I hope that audio is going well and that everybody has power around Melbourne and elsewhere despite the Darwin-like weather. Here at the Institute, our staff and our students and our board celebrate and acknowledge the traditional owners and custodians of the land on which we meet today. And here in Parkville, in our Wehi building, that is the Wurundjeri people of the Kulin Nation. And I'd like to pay my respects to their elders past, present and emerging, and the elders from other communities who might be participating in this COVID-19 panel event today. And I'd also ask that wherever you're sitting, that you just take a moment to acknowledge the traditional owners of the land where you are. Hello and welcome to this evening's special event to hear about Wehi's COVID-19 research and for the opportunity to ask our panel of wonderful experts any questions you might have about COVID-19. Honestly, I did not think in February 2020, when the news of this pandemic was just breaking, that 2 years in 2 years' time, we would still be having challenging issues that we're confronting as a community. In the past, when you've heard me speak at Wehi Research Fora, where we talk to our wonderful stakeholders and supporters about what we're doing, you'll often heard me talk about research being a roller coaster and the fact that you can go for days, weeks, and sometimes months or years where your experiments don't work and where the support of your colleagues and your support of people in the community is what gets you through those difficult times. I guess as I reflect over the last couple of years, my enduring feeling is absolutely being on a roller coaster. And you can see the background behind me, the different phases of the pandemic, and that's case numbers worldwide. And you can see that the graph absolutely looks like a roller coaster. I think the impact of on all of our lives has also been a roller coaster. And I want to again acknowledge the resilience of the staff here and the students here at Wehi, but also the resilience of our whole community and the support that we have found so beneficial from all of you. This evening's event is going to be recorded for those who can't attend, and we will send a link to the recording to everybody who is registered in the coming days. And obviously, if you have friends or family that you think will be interested in the discussion tonight, this evening, share that link with them too. Today, we have a really special panel of scientific experts to talk about COVID-19 and some of the really exciting research projects Wehi has underway to combat this disease, as well as combating future pandemics and infectious disease outbreaks. So I think much of the work we're doing is both as a response to the current pandemic, but to ensure we're better prepared next time. Our speakers are going to provide an overview of their research, and following that, we will open the forum up to you to ask questions. And I'm sure you are all experts on online fora, and the questions can be submitted at any time via the Q&A box on your screen. Please try and avoid using the chat functions to ask questions because we might miss them. In the Q&A box, you can also vote up questions that you think are exciting. And those with the highest number of votes will be the ones that we get to first. As often happens in fora like this that generate a lot of interest, unfortunately, we may not have time to answer every question today. But what we will do is our best to respond to any unanswered questions in the coming days. Just before we get started, you will see that a poll has appeared on your screen. Could you take a few seconds to answer the questions? Because one of the things that we try to do is to ensure that the events that we create are aligned with what you are interested in. And one way for us to understand that is to get feedback from you. So if you could fill in these questions as I am going to be doing now, we would be really grateful. Thank you all for that. That will be really useful in terms of tailoring future events. It's really hard to believe we're approaching the second year living with COVID. I've really missed the meetings face to face, but as vaccinations become more widespread, and I think as a state, we're heading pretty rapidly to a 95% double dose level. And as we learn how to live with the virus, there is a sense of normality starting to return to everyday life. But as I said, it's been a roller coaster. And just as I thought things were returning to normal, we've of course had the emergence of a new variant of concern that we have to be wary of. And I know, like you, the announcement by WHO of the Omicron variant was pretty daunting and disappointing. And I also know that it's still very early days yet, and there is a huge amount that we don't know about this new variant. And I think that not knowing also adds to the anxiety. What we do know, though, I sound like a politician, what we do know and what I can tell you is that there are some of the best scientists and health professionals in the world investigating how this new variant will affect us. The emergence of Omicron is a timely reminder that we will be living with this virus for some time to come. And it's vitally important that we continue to invest in research. We can't take things for granted. To assist our scientists in understanding how this virus and other viruses work, we need to invest in new rapid detection methods and new treatments and new improved vaccines. And it's through medical research that we will be able to tackle not only the new variants that cause COVID-19, but also ensure we're ready to face any future pandemics. The research that you're going to hear about tonight and all of the amazing discoveries that happen at WEHI could not be done without your generous support. While medical research is immensely rewarding and a thrilling occupation, it can be really difficult, as I've said, a roller coaster. The ever-present challenge of securing funding for important research is daunting, and it can be discouraging. Your support provides WEHI scientists with not only the financial resources to progress their important research, but also, as I've emphasised time and time again, so much encouragement. I know firsthand what a huge injection of inspiration that your support can provide. Whether you've donated to our appeals, established a scholarship, support a research centre, or raised money through a barbecue, we are exceptionally grateful. And I want to thank you on behalf of our staff, students and board. And I look forward to being able to thank you in person in 2022. Tonight, you're going to be hearing from three panelists who are expert in their fields. Professor Mark Pellegrini, who is Joint Division Head of Infectious Diseases Division and an infectious diseases clinician, is going to give you an overview of where this pandemic started, where we are now, and he will talk about the new Omicron variant and what we know so far. Mark has also done a fabulous job throughout the last couple of years communicating issues around COVID to the public through numerous radio interviews. And I just want to acknowledge also his guidance to the leadership of the Institute as we've navigated the pandemic in order to both protect our staff and students and their families and communities, but also contribute to the research that's going on about COVID. Dr. Vanessa Bryant is the second of our panelists, our experts, and Vanessa is a laboratory head in the Immunology Division, who is leading the COVID profile study, which aims to understand how immunity to COVID develops, how long this immunity lasts, and what happens when the immunity is lost. And finally, Professor Ivo Mueller, who's Joint Division Head of the Population Health and Immunity Division. Ivo's working closely with Vanessa on COVID profile, but he's also supporting the health authorities in the Pacific with modelling COVID-19 infection scenarios. Before we get on with hearing from our scientists, I also wanted to touch on a couple of other areas WEHI scientists are investigating in response to the pandemic. Very excitingly, last month, we announced a partnership with CSL called the Centre for Biological Therapies. One of the exciting projects underway is the production of monoclonal antibodies to combat the SARS-CoV-2 virus, which causes COVID-19. This research has been led by Associate Professor Weihong Tan and has shown that monoclonal antibodies can block the SARS-CoV-2 virus from entering a cell. Blocking the virus from entering the cell means the infected individuals have a lower viral load, will recover more quickly from an infection, and be less likely to pass it on. These biological medicines are vital as they will be frontline treatments for the most vulnerable people, and unlike vaccines, these therapies provide immediate protection. In the future, we'll be able to use these treatments in the immunocompromised and those unable to be vaccinated. Second project I wanted to touch on is around drugs to treat COVID-19, which are also a focus of WEHI researchers. Using the National Drug Discovery Centre, which is based at WEHI, researchers have tested 40,000 new compounds against SARS-CoV-2. They identified several potential candidates and are currently assessing their viability to use as a COVID treatment. Still early stages of discovery, but as this virus continues to change, this research is preparing us for the next outbreak. While increasing the range of treatment options remains incredibly important, the development of better rapid testing methods is crucial for containing outbreaks, and WEHI researchers are also working on diagnostics. Our goal is to develop faster tests for infectious diseases, including COVID, so that people can be diagnosed very accurately on the spot within minutes rather than hours. These rapid screening tools could be used in hospitals, doctors' offices, and airports, and infected people could be identified, isolated, and treated immediately, preventing disease spreading further. WEHI researchers have also been instrumental in wastewater monitoring that's been so helpful in alerting contact tracers to potential outbreaks. Wastewater management is an invaluable tool alongside testing and informs health officials to increase community testing in areas where positive results have been found in wastewater. So now on to our scientists who are here to answer your questions about COVID-19. And just a reminder again, submit your questions at any time in the Q&A box. Over to you, Mark.
Thank you, Doug. I'd like to begin by just sharing a few slides and particularly trying to explain why we see variants like this Omicron that we're currently facing right at the moment. So hopefully you'll be able to see my slides now. And the first thing that I wanted to try and explain was why do viruses actually change? So viruses are made up of nucleic acid, the building blocks of our chromosomes, albeit this particular virus has got a slight variant as opposed to the nucleic acids we've got in our chromosomes. It's made up of RNA rather than DNA. But viruses change their genetic codes to evolve and to replicate better. So in the case of SARS-CoV-2, if people all start wearing masks, it needs to adapt to that and will try and find a way to be transmitted better, to latch onto ourselves better, and to actually be expelled better. And so that's what this virus has done when it changed to the Delta strain. It actually was able to latch onto ourselves a bit tighter and actually produced itself a bit better in the cells that got infected. Viruses also adapt to our immune responses. So our bodies are built to recognize viruses and prevent those viruses from actually infecting cells. So part of that immune response is the antibodies that we produce, either because we're naturally infected or because we're vaccinated. And again, the virus needs to adapt and change its coat so that it's no longer recognizable by our immune system. Viruses also tend generally to adapt to changes in human behavior. So if people are very sick, very early on, we started to quarantine and isolate those people. So viruses, for the most part, and at some stage, SARS-CoV-2 will do the same. It will modify such that it doesn't cause severe disease, because if people start getting a lot of symptoms, they'll be isolated, taken out of the population, and the virus can't transmit so well. So generally speaking, this is why viruses try to change their genetic code and evolve, primarily so they can replicate better and transmit from person to person better. Now, in the case of SARS-CoV-2, it's got this spike protein, and this spike protein, which is on the surface of the virus, is what clamps onto our cells. And because that's such a vital part of the virus and determines whether it can attach and enter into the cells, that's the one that's most prone to mutating. And you can see in the next slide, this figure's been covered, but it's an electron micrograph. Here are the actual virus particles. And as I said, on the surface of those viral particles is this spike protein, which I'm calling the clamp, that hooks onto our cell, which is shown here. And it clamps on quite strongly, but as the virus has evolved, it's clamped on stronger and stronger so that it can enter cells better and be transmitted better. Now, when we see this virus, our immune system will make a plethora of different responses, but one of them is to make antibodies. And these antibodies will try and hook onto this clamp that the virus has got and prevent that clamp from working and attaching to our cell membrane. So there's a couple of reasons why the virus will want to try and change that coat and change that spike protein. One, to hook onto our cells better, and two, to avoid antibody responses that we're making. So here you can see, maybe we can call it the Omicron virus. It's changed color and it's changed spike protein. And it's done that to hook onto our cells better and to avoid these particular antibodies from binding. Now, we've had many variants of concern, and you can see here some of them detailed here in this WHO work. So most of the variants that we've seen over time have changed the spike protein a little bit. So you can see in the Delta, which is now the world dominant variant of SARS-CoV-2, had around about 4 or so mutations in that spike protein that conferred a real advantage to it compared to the original UK variant, which was the Alpha strain, and indeed the Victorian variant. But the thing that was sort of like made people a little bit alarmed was that Omicron actually has got well over 30 different mutations in this spike protein. We haven't seen that before. You can see some have had 5, some have had 4, but we've never seen this many mutations. We don't know what it means, but they're all in the spike protein and they must be there for a particular reason. But Omicron's not been the only one, nor those others. We've had many, and some of them have sort of fallen by the wayside, and that's what I'll go through in the next few slides. You can see Mu that was in Colombia, but quite a few others that have happened over time. And so they still are variants of interest. Others have sort of become variants that we just monitor. And on the next slide, you can see a whole lot of variants which have actually been de-escalated, or rather, we don't think they're that important anymore. So I don't expect you to read any of these slides, but it's just to show you the sheer number of different variants that we're dealing with over time. So the Omicron virus it's a cause for vigilance, but not a cause for alarm at this stage. Does it transmit better? Probably, but we still don't have conclusive data to tell us that. But all those mutations in the spike protein probably amount to that virus hooking onto ourselves a little bit better. There's certainly preliminary evidence to suggest that people who have been infected with COVID in the past will now be able to be infected with the Omicron variant, meaning that naturally acquired immunity might not be enough to prevent you from getting infected with the Omicron variant. It's still quite unclear if our current vaccines will be quite as effective as they have been, or if they'll be compromised. But what I can say is that our vaccines will definitely remain effective at preventing severe disease and death, even with Omicron. So our PCR tests also might need to be adapted, and that's not completely clear. PCR testing is probably going to be quite adequate, but the rapid antigen tests that have flowed into the market recently, some of them might not be as good at detecting Omicron. It's certainly unclear if the monoclonal therapies, the biologics, will remain effective as well, because Omicron has changed that spike protein so much. But some of our therapies will remain quite effective. So molnupiravir, which actually stops the virus from replicating, not licensed here in Australia as yet, but probably will in the time to come, that will remain effective. And indeed, a lot of the research that Doug talked about in terms of developing new antiviral drugs based on a national drug discovery scheme, those drugs that will be developed won't really be affected at all by changes in spike protein, because they're really trying to target the ability of the virus to replicate. And indeed, what WEHO has done quite ingeniously is it's progressed two arms to attack the virus, and the combination of those two arms will make it very, very hard for the virus to evolve. So that's the current state of play, I suppose, with the Omicron variant. And we're certainly trying very hard to create therapies that will be pretty much refractory to a lot of these new variants as they come up. So I'll hand over now I'll stop sharing my screen, and I'll hand over to Vanessa to talk about the immunology of COVID-19.
Terrific. Thanks, Mark. Just wait for you to stop sharing, Mark.
Sorry, it takes me a little time.
I think it takes us all a bit of time. We all need a bit of time. So thanks, Mark. Good evening, everyone. I'm very happy to be joining you today to tell you about our COVID profile study. So the COVID profile study, I actually co-lead it with Ivo Mueller, who will speak in just a moment. The team actually expands 8 labs across WEHI and the Parkville precinct, and together we've got, as you imagine, a diverse expertise that spans across immunology, infectious diseases, epidemiology and modelling, as well as genomics and clinical care. We launched our COVID profile study over a year ago in October 2020. It was actually under lockdown 2, which seems like a long time ago now. One of the goals of our study was to determine what is necessary for long-term immunity to COVID-19 and if that's even possible. We wanted to be able to rationally plan for a safe and sustainable path to fully reopen. And we recognised that this is absolutely dependent on knowing what immunity to COVID-19 looks like, how long it lasts, and who is most at risk of reinfection or poor recovery. We've also recently expanded our efforts to determining the effectiveness and timing of booster strategies, as well as, of course, immunity to emerging variants of concern. So in our study, we've enrolled 100 adults in Melbourne who have recovered from COVID-19 and over 70 healthy volunteers who never contracted COVID. And our study volunteers generously and regularly give blood and serum samples to our research. And we're also following their responses as they receive their vaccination and now as we look towards boosters. So in the first phase of our study, we tracked the virus-specific antibody responses over time in people recovered from COVID. And we showed that most people still had durable antibody responses, but for a small proportion, 12% of recovered volunteers actually had no detectable specific antibody levels between 6 and 12 months after they recovered from the SARS-CoV-2 infection. Now, this doesn't mean that they don't have immunity. Just because we don't have antibody levels doesn't mean we don't have immunity, because while antibodies are an essential part of immunity, in a normal immune response, antibody levels in our blood naturally wane over time. But our immune system also generates specialized memory B cells and memory T cells that patrol in our circulation. So if we encounter the same virus again, we can very quickly make a large amount of protective antibody and immune cells to rapidly fight infection and prevent severe disease. So one of the studies currently underway in our group is where we directly assess these rare virus-specific memory cells in the blood to determine in how many people can we detect these rare cells, for how long do they persist, and how much protection do they provide. And together, these studies will allow us to identify biological signatures that correlate with protective or waning immunity. One of the other aspects we're investigating is long COVID. So within the first few months of launching our research, we realized that about a third of our recovered volunteers were really struggling with persistent symptoms. And these were symptoms that are not necessarily associated with post-viral recovery, such as shortness of breath, memory loss, brain fog, fatigue, heart palpitations, ongoing muscle and joint pain. And we now, of course, know this as long COVID. And now, almost 2 years on since the virus first emerged, the definition of long COVID is still very much evolving, and there is no standard treatment. And we found in our study that these symptoms can persist for 3 months to more than a year in some people. And we work very closely with the clinical team at the COVID Recovery Clinic at the Royal Melbourne Hospital to track recovery in this group of participants, as well as any reported changes in their symptoms, and link this with our immunological studies to understand what is driving long COVID, who is most at risk, and how can we directly target its underlying cause. And the third arm of our study is really focused on vaccines and boosters. So comparing the responses in people who have recovered from COVID to healthy volunteers who were never infected will give us early insight into the effect of boosters, how long a boosted immune response is sustained, so we can rationally inform the timing of future booster vaccine strategies, as well as also identify vulnerable groups with waning immunity. And because we have a really broad range between the timing of when our study participants were infected with the virus and when they were vaccinated, this gives us insight into the optimal timing of booster intervals for long-term protection, as well as, of course, protection against the variants of concern that Mark spoke to. So from our results so far, we observed, as expected, a really large boost in virus-specific antibody levels after vaccination. And overall, this is even greater in the COVID-recovered volunteers. And while we're still collecting data for the duration of immunity post-vaccination, we've tested how well these antibodies actually neutralize the virus, both challenging against the original SARS-CoV-2 virus strains, as well as the variants of concern, which does include Delta, but not yet Omicron. So overall, we observe a solid neutralizing activity against both the original strain and against the Delta strain. And our early results are suggesting that these responses may be enhanced in people who were previously infected. So this really fits with what we're seeing in the real world data. So although we are seeing some breakthrough infections in vaccinated individuals, they still make up an exceptionally small proportion of hospitalized and ICU cases. And finally, I just will note that one of the questions we sometimes get is, why do this research in Australia where, relative to the rest of the world, we have so few cases of COVID? And I will say that this is actually exactly why we need this local data. So here in Australia, we're really one of the few countries that has successfully managed to avoid widespread community transmission before our COVID vaccine rollout. So unlike the rest of the world, most people in Australia are actually immunologically naive before we were vaccinated. And we now have one of the highest vaccination rates globally. And what that means is that we have a slightly different immunological landscape to many other countries where COVID research is happening. And these countries with high levels of transmission that can act as a continual boost once immunity is established, and where vaccination may act as a further boost to existing immunity. So, you know, we must always be led by the science and by the data generated globally as a collective, and this has been exceptionally informative. But we also have to look to our local data and design our booster and our public health strategies on a combination of both. We're very happy to contribute local data and feed our findings into modelling strategies to predict the timing and frequency of long-term booster strategies in Australia. And Ivo will talk about some of that work now. So now I'll hand over to Professor Ivo Müller, who will talk about other research efforts as well as data modelling. Thanks, Ivo.
Thank you, Vanessa. It's a great pleasure to present some of the work we've been doing in collaboration with many friends and colleagues around Melbourne and elsewhere in Australia on modeling COVID-19. I have to quickly apologize if my sound may be a bit bad because we're just being hit by a storm. So if it gets too bad, please let me know if you can't hear me properly. So how did we get into modeling COVID-19? So in my group, we have been modeling malaria in different parts of the world for the last 5 or 6 years. And we have joined with colleagues in Melbourne and in Canberra and in Queensland on a project to help build capacity for modeling different infectious diseases in our neighboring Asia Pacific countries. That's a project supported by the Department of Foreign Affairs and Trade. And we've been doing that, and malaria was supposed to be our case study. And that project started in early 2020, and obviously when COVID-19 hit in March 2020, almost our entire program very quickly pivoted from malaria and flu to COVID-19. We then used our knowledge that we had from modeling malaria to then basically build a model to look at what transmission of COVID might happen in our neighboring Pacific countries. And we applied that model to help authorities in PNG, French Polynesia, Fiji, Tonga, and a couple of other countries plan their health systems and see how they could strengthen their health system for the arrival of the virus. And that was work that we did on behalf of WHO and the Western Pacific Regional Office in Manila. In May 2021, our group, led by Professor McVernon at the Doherty Institute, was asked by the federal government to provide modeling to support a national plan for reopening Australia. And because we had our models for modeling transmission for the Pacific, we then joined the consortium and started building models for modeling transmission of COVID-19 in Australia. So I think I want just to dispel a few assumptions maybe that people have around the Doherty National Plan model. The first thing that is really important to point out, this is not just the Doherty Institute. The team that has been part of that modeling that has supported the federal government's and also state government's plans for reopening is a team of over 30 people that work in 12 different institutions in Victoria, New South Wales, South Australia, and Western Australia. So it's a real collaborative effort of almost the entire Australian modeling community across many different institutions. And we are very grateful that we at WEHI have been asked to join this. The other thing that is important to understand is that it's not a single model. It's actually a whole suite of models that do model different aspects of the pandemic and help leverage that knowledge to then help policymakers make the optimal policy settings. So in particular, we have models that try to evaluate and predict the impact of different interventions and different mixtures of intervention on the abilities of the virus to transmit. Then we have a model that then allows us to predict, based on a certain setting of public health intervention, what the expected number of cases looks like and what incidence of COVID infections we see. And that's the modeling that we've been doing at WEHI. That's our contribution to that national plan modeling. And that data then gets fed into other models that then predict how many people may get severely ill, may get hospitalized, and what the risk of death might be in a context of different vaccination levels in different intervention settings. The other thing that's very important to understand is that we are not modeling the future. We're not trying to predict what's happening in March 2022 or in June 2022. We're modeling what we're calling scenarios. It's the question of what would happen if we do one thing or we do the other thing. That helps policymakers to make the decisions what they might want to do and what they might want to introduce in terms of public health measures. Obviously, the introduction of those public health measures and the evolution of the virus, as Mark has said, then changes how the virus actually reacts and how the pandemic actually then proceeds. And so the models will always be wrong because the reality changes. Now, all that work that we've done was done on behalf of the Department of Prime Minister and Cabinet and the Federal Health Department. So we've really worked for the federal government and not so much for the state government. But all these results were presented to National Cabinet as well, which has made it quite difficult for us to talk about it in public. And that's why there hasn't been so much information out there, because a lot of that information is protected by cabinet confidentiality and therefore cannot be shared really until it's been approved for release by the federal government. However, the main reports of the modeling that has been released so far are actually available on the Doherty website. So what have we done? Obviously, we have contributed to the famous 70 and 80% and 90% thresholds that have underlined the opening strategies of the different states. But a lot of further modeling has done, both in terms of looking at the sensitivity of these thresholds based on how many cases are circulating. We also did significant work in trying to refine the test, treat, isolation, and quarantine protocols to make sure we do the right thing, that is giving us the most impact and not wasting a lot of effort into chasing up things that are not so useful. We have looked at questions in special populations like First Nations people, or what happens in school, and what should be the optimal strategies of keeping schools open and keeping cases low as possible. Another set of modelings has looked at border measures and looked at the issue of hotel quarantine versus home quarantine and different testing strategies associated with different forms of quarantine. The work we are working on right now is around booster strategies. So we are working, and that is actually a project that is co-led by us at WEHI with colleagues at Uni Melbourne and the Doherty Institute. And that's really to start looking at the questions of how many boosters do we need? When do we need boosters? So the question is, how durable will vaccine protection be in the population? Because as Vanessa has mentioned, immunity, naturally acquired and vaccine immunity, tends to decay over time. What is the optimal interval for a booster? When should we get a booster? But also really important, what level of uptake of boosters do we need? How many people do need to get boosted in order for us to really achieve the public health impact that we're looking for? And in this context, we are also using some of the data from our local studies, COVID profile, and our studies. Obviously now with Omicron coming, we are in the process of also pivoting some of that work to look at what the potential impact of Omicron arriving in Australia would be on the risk of cases in Australia, and what would be the optimal strategies of combating Omicron if it does reach our shore, if and when it does reach our shore. So thank you very much. That's a quick overview of what we've been doing on the modeling front.
Thank you so much, Ivo, Vanessa, and Mark. That is fantastic. Just let me get myself organised here. Now what I think we can do is go to questions, which I have here, and I'm going to read them in order of your popularity of them. First question from an anonymous attendee. How do you explain the high number of COVID cases in Victoria versus the other Australian states? Might ask Mark to answer that first. Mark, maybe our panelists can pop their cameras back on so I can see them. Mark, how do you explain that one?
Yeah, I mean, I think that the modelling, and this comes back to Eva, always predicted that we'd be up into the thousands and several thousands when we started to take away the rules that were constraining transmission of the virus. And it is taking a little bit of time. In terms of why are we so different from Sydney, well, there's probably many rationales. They got vaccinated a little bit earlier, a little bit more quickly, but also they probably have fundamental different socialisation networks than we do in Victoria. There's different populations and demographics, so we know that some of our communities are less vaccinated than other communities. So I can't give a hard and fast answer, but we're not a hugely different, we're not hugely different in terms of the number of cases we're having here compared to the number of cases up in Sydney. And obviously the other states have got smaller populations, less densely populated than Melbourne, and never had that super introduction that we had here. But Ivo, did you want to add a comment?
Yeah, so it's hard to predict exactly why, and say I think I would also support what Mark says, that we're really in the range of cases that we have predicted. I think compared to New South Wales, the fact that New South Wales did get an additional boost of vaccines early on and has used those vaccines very specifically to target their hotspots, that has made an impact. I think that does explain partially why that those extra vaccines very targeted against high transmission areas has probably reduced their numbers a bit faster than ours. I think the the lockdown fatigue in Melbourne has probably also led to the fact that we as Victorians have probably taken our freedoms a little bit quicker and maybe followed rules not as accurately as the New South Wales people did for the time when they were in lockdown. And so our lockdown may not have been as effective, and therefore the virus got disseminated more widely in the community, and therefore we're seeing a slightly higher rate of ongoing transmission.
Can I ask a slightly more optimistic question? Maybe I'll put the question differently. How much should we beat ourselves up about the transmission in Australia compared with other places? Have we generally handled things well? Is there and I understand there have been many different difficult impacts on people, from the individuals whose families have passed away, family members have passed away, people that have become very ill, people that have had impacts economically. I understand that this, we shouldn't be patting ourselves on the back too much, but equally, should we be beating ourselves up too much about our poor handling of the pandemic?
Oh, so I was just thanks, Doug, for that intervention, because I was just going to come to that point and basically say, what we also have to see is that we really see a stable number of cases in the context where we've dropped almost all restrictions. So we do not see the virus growing. We see cases stable, which means we're actually doing a really good job with the vaccinations and other things we've done. And also the way we have opened up is really exceptional compared to other parts of the world. So as I completely agree, I think the numbers in Victoria are a little bit higher than in New South Wales, but they're very, very low internationally. And the reproductive rate of the virus is 1 or below. So they're, despite massively opening up and giving a lot more people moving and meeting and all those restrictions, the numbers are going down or are stable. So yes, I completely agree with you, Doug. We're doing actually exceptionally well.
Very good. I've got a wonderful question from Ian McFarlane, one close to my heart because I am in exactly the same position that Ian has. Ian says, I am double vaccinated with AZ and are due for a booster in January, which I think is the same time, Mark, that I need my booster, having got my shot in July. Which vaccine should I choose for my booster and why? I would ask you, Vanessa, but I think this is medical advice. So maybe you and I and Ivo are less qualified to give it. Mark, and then Vanessa, if you want to correct Mark, I'm still happy as an immunologist for you to correct him.
Boosters will predominantly be Pfizer vaccine, and there's some rationale around that. But that's what's currently being rolled out as the booster. If people can't have the Pfizer vaccine, then the other vaccines are certainly available, being AstraZeneca and the Moderna vaccine. But Pfizer is currently what's being rolled out as the booster.
And another vaccine question that I have, Mark, what's the Novavax? We kind of heard this whispered, and some of my family members that are more vaccine hesitant have been holding out for this Novavax thing. Tell me what's going on there.
Yeah, I mean, I think that certainly with the AstraZeneca vaccine and the Moderna and the Pfizer vaccines, they're relatively new technologies. AstraZeneca are a little less so, but we haven't had many vaccines of that type, which uses another virus to introduce the spike protein of COVID-19, or rather SARS-CoV-2. And the mRNA vaccine is going to revolutionise vaccination, but obviously there's a degree of hesitancy about taking up a new type of vaccine. Novavax is much more of a conventional method of creating protein and vaccinating people with the coat protein of the virus.
So old tech.
It's sort of old tech.
Like
but obviously there's been some obstacles in elements of production and so on, which have delayed things, but it is sort of more the old tech.
Okay.
And some people are much more comfortable with having an old tech vaccine.
A brilliant question here from David A. That is for Vanessa. Vanessa, are there any discernible trends in the long COVID data which suggest risk factors, e.g. age, lifestyle, obesity, pre-existing conditions, and I would add into their ability to make an immune response? What's going on with long COVID? Why do some people get it and some people don't?
Well, the second part of your question, we don't know yet. We don't know why some people get it and some don't. Look, there aren't really discernible trends on who's more susceptible to long COVID other than people who have had COVID. And we know there are susceptibilities in terms of age, people who are immunocompromised, some people who have comorbidities and pre-existing conditions. But outside of that general COVID susceptibility, there doesn't seem to be really strong links. We're part of a Victorian-wide long COVID research group of clinicians and researchers trying to look at the data in every which way. We're starting to see that, with more and more numbers, we are seeing more people who were hospitalised do have long COVID, but it's nothing really overt. We're seeing people even with mild disease, young people, and the symptoms that people are experiencing are so incredibly diverse. So it's entirely likely we're looking at many different types of disorders within that. So it'll take a while to unpack yet.
And do you think we have the tools and techniques to unpack it over time as we begin to understand it? Are you optimistic?
Yeah, I think so. And I think it will also provide big insight into other chronic diseases that are really hard to measure. One of the things about long COVID is that our understanding of it really emerged from patients saying, this is my experience. I'm really struggling going back to work. I work a shift and then I'm in bed and I can't get up. And this is the experience of lots of other people with different chronic diseases that we don't think necessarily have an immunological basis. They're really hard to measure in the clinic. They're really hard to understand. They're really hard to treat. And, we do have some front runner theories about, what might be the link with the immune system with long COVID. One theory is that, maybe there's some non-infectious viral fragments that are persisting. Maybe it's a hidden reservoir where we can't detect diagnostically, or maybe it's this immune misbalance where the immune cells are still misfiring. There was some early evidence that maybe vaccination can help alleviate some of the symptoms, and that provided some early cues that maybe the immune cells are either mopping up some residual virus or actually redirecting the immune cell responses to be more targeted. But it's still early days, but this is something we're actively investigating.
Fantastic. We've got a great question from Kerry that I'm going to ask Ivo. Hi panel, is there any commercial test available for checking antibody levels post-vaccination? I've heard friends in Europe talking about paying for this test. Do we have it in Australia, Ivo?
So there are commercial tests for measuring antibodies. They are generally for reference laboratories. So they have been around for quite a long time, where big pharmaceutical companies have made standard tests that have been endorsed by WHO as well. Those tests are available in major diagnostic centers. Whether they are available to individuals to be able to go and get their antibody tests without having them ordered by a doctor, I would have to refer to Mark and the clinicians, but those tests are available. What are not available in Australia are rapid tests for where you can go to the pharmacy, you get a rapid test like a pregnancy test and can get your own antibody checked. And that's because those tests for antibodies, those rapid tests are not as accurate as tests for the virus. And therefore, that's why they have not been approved by TGA for use in Australia.
But are kind of antibody tests going to be used to determine who might need boosters, number 3 or number 4 down the track, Mark? You're muted.
Yeah,
you sighed. I could hear you sigh. Did I ask a stupid question?
No, the sigh was I have to unmute myself first.
Okay.
You need some artificial intelligence to detect that my name goes on and therefore I immediately get unmuted. The immunity is more complex, I suppose, than just antibodies. So, and then antibodies themselves don't tell the whole story. And then antibodies may be relevant to one variant and not to other variants. So I suppose being able to decipher from a simple test whether you're going to be immune to the next variant would be very tricky. And to be able to say, well, you need exactly this level to be immune from SARS-CoV-2 is also tricky. It's pretty much a sliding scale, and It's not, strictly speaking, going to be incredibly accurate to be able to inform people as to what they should be doing.
Another wonderful question from the equally wonderful Jenny Tatchell, and she asks, would the Spanish influenza have been as deadly if it was around today? That's her first question. Or is it still around? Is our present influenza vaccination similar in action to the COVID vaccination? Lots of deep philosophical questions there. I might ask Ivo first. If Spanish flu hit today, Ivo, you're a modeler. Give me your prediction.
So in terms of it being as deadly, I wouldn't be having such a high death rate because we are much better at treating severe cases. We have much better health system. We're much better employed by ICUs. We have antibiotics now, and in 1917 we had no antibiotics, and people who got secondary bacterial infection, secondary bacterial pneumonia would die at much higher rate than they would be dying today.
And of course, we have Relenza.
We have Relenza as well.
WeHi's own Peter Coleman, living legend, while he was at CSIRO.
Yeah, so we have antivirals as well. In terms of the vaccination, obviously we have the flu vaccine, and I think we should point out here that our COVID vaccines are actually better vaccines against COVID than our influenza vaccines is against influenza. And that's partly because influenza mutates so quickly, and we always are vaccinating against last year's influenza strains and not against the new influenza strains. And that's why the protection you're getting from an influenza vaccine is maybe in the range of 60 to 70% protection, while the COVID vaccines, in particular against severe disease, give us a much higher protection, give us a 90 to 95% protection. So I think I think the first answer is no, it wouldn't be as deadly as it was in 1917 because our health systems are made better prepared. And the vaccination is similar, but I think actually our COVID vaccines are actually probably better than our influenza vaccine.
Very good. And the Spanish flu is not around every year, but we did have, we held our breath a few years ago. Was it 2009? Was that Spanish flu or was that a different variety? Was it, I can't remember what it was, H1N1 or something like that? Mark?
Yes, H1N1 a few years ago, but it just wasn't particularly transmissible, and it turned out to be nothing of major concern. But yes, the flu will change its colours from time to time, but we'll always have this background immunity that will confer something to us, even though it changes its colours a little bit. But I think we will be able to respond to it much, much better now that we've had experience with SARS-CoV-2. We know we can shut borders. We know we can stop international travel. And if a bad flu came around, we'd be able to respond very quickly.
Very good. A fabulous question from Pat and Ruben Urban. Are any of Omicron's many mutations in the areas which the mRNA vaccines target? How would this affect getting a booster? Mark, you are still off mute. If you don't know the answer, handball that to someone who does.
Yes, so there's all likelihood that it will marginally affect the vaccines. Delta did, so we knew that the protection against infection with Delta was reduced, but it didn't really impact the ability of the virus to prevent severe disease and death from Delta. So I still feel that even if the many mutations in the spike protein impair the vaccines a little bit, they'll still be quite effective in preventing severe disease and death. All that work is still being done. So obviously everyone's rushing to understand if the vaccines will still work as effectively as they did with Delta. But that work is being done urgently by many, many scientists around the world.
And I'm going to ask a supplementary question. If it turned out that a variant arose overseas that was escaping our mRNA vaccines, our vaccination, how long would it take us to manufacture a new one, a replacement?
That's a great question, and I'd say that
I know. That's why I asked it.
Theoretically, incredibly quickly. So you could do something overnight. But that's theoretically, because all you have to do is change the code in the mRNA vaccine and off you go. But it does require a whole new manufacturing process, and then there's a distribution process, and then you got to get everybody back in to get that booster shot. So there's a big difference from doing it on paper and actually pragmatically getting everyone vaccinated.
And I should declare that I was asked to be on the mRNA working group for Victoria, the Victorian government, and I do think it is exciting for us to be able to have the ability to manufacture those vaccines in Victoria and therefore make us less dependent on overseas supply chains. I think that will be really important should the pandemic continue, but also for what will be, inevitable outbreaks of other infectious diseases, whether in Australia or in our region. I think that's a really exciting step forward. Okay, an anonymous attendee is asking a question I've heard many people ask, and that is, has WeHi tested ivermectin against COVID? And I'm going to get Ivo to answer this. Tell us what ivermectin is, Ivo.
So ivermectin is a drug to clear helminths and other parasites and scabies. And it's predominantly an animal drug. It's a veterinary drug that's used to clear helminths and gut infections from cattle and other veterinary indications. In humans, it's only really used, and it's not licensed for that, to treat scabies, which is a skin mite infection. And that's what it's been used for in humans. There is very limited evidence, or rather no evidence from clinical trials, the ones that have been conducted, that it has any effect against COVID-19. So there is strong evidence out there that it is not effective to treat COVID-19. And we at WeHi have not tested the effect of ivermectin against COVID-19.
Thank you. A question from Kim Burtonbrighter. Are people with long COVID still infectious for the entire time they have the symptoms? Another brilliant question. Vanessa, give us a brilliant answer.
Thanks, Kim. We don't think people are infectious for the entire time they have long COVID. Different people have different infectious periods, but they don't really extend to the 3 months, 6 months, 1 year, the sorts of timelines we're seeing with people with long COVID. And that is because we do think they're either non-infectious viral fragments or really small reservoirs that are not sufficient to be actively replicating and infecting other people.
Fantastic answer. And panel, are you happy to go a little bit longer? There are some fabulous questions, and I know there's a real appetite for expert answers. Mark, you're not going on the catwalk to show off your shirt or anything like that? Nothing? All good? All right. I have another great question from Ian McFarlane. This is a really important question. At what level in the general population do we reach herd immunity? And at this point, can the unvaccinated be allowed to participate in everyday life without restriction? Ivo, give us an answer. What's the magic number? Surely there is only incremental gain.
So there is no magic number, unfortunately, because in COVID-19, because immunity is transient and is not lasting forever and decays over time, there is not a herd immunity like we get in measles, where the immunity will just simply stop transmission and transmission will no longer occur above a certain level of vaccination. Also, vaccination in COVID is very, very good against severe disease, but it does not stop all transmission. There are still low levels of transmission happening even in vaccinated people, which means unvaccinated people remain at risk of being found by the virus and having severe disease, no matter what the level of immunity and vaccination is in the population. And transmission will also continue because the vaccine does not stop all transmission. And I think that therefore the unvaccinated people will remain at risk of the virus and severe disease as long as they remain unvaccinated.
So, Ivo, is there another question? And that is, at what level in the general population do we reach before we run the risk of overwhelming our healthcare system? And at that point, should unvaccinated people who've made an informed choice not to be vaccinated be allowed to participate at their own risk in society? What's the magic number for the healthcare system?
The magic number for I can't tell you what exactly the magic number is. I think it is somewhere in the range, probably in the range of 90 to 95%, where we would feel comfortable that the health system does not get overwhelmed if we have everybody participating. And that's in a sense what's also happening in some of the states here. So I think that the numbers for the health system, I think once we have that high rate, look positive. But that also does require that the people who are vaccinated are getting their booster shots.
Absolutely.
Because that is required to maintain that high level of protection. And if the people that are vaccinated are not getting their booster shot, the numbers of cases will go up, and then the health system is at risk of being overwhelmed. So therefore, the message clearly is to everybody, yes, you can make an informed choice of putting yourself at risk by not being vaccinated, but we would really encourage everybody to get vaccinated, and we would encourage everybody to get their boosters as soon as they're eligible.
Mark, happy with that answer? Any arguments with Ivo on that one?
No, I mean, the only thing that I would add is that, you know, if someone's unvaccinated, then you're making the informed decision for yourself that you're comfortable with the possibility that you'll get severe disease and the possibility that you might die. But there's also the impact that you might have in getting infected and then passing it on to someone else. So there's going to be still a lot of people in the community that will remain vulnerable. And those people will be those that have got a compromised immune system, people with cancer. So, you know, the decision around yourself is fine, but there'll always be this added, I suppose, guilt around, well, I could be a vehicle of transmission if I haven't been vaccinated, and I could be causing disease for others.
And what is the difference in the rate of transmission between a freshly vaccinated person and an unvaccinated person? It is significant. It's not absolute, but it is an important element of our public health response.
Oh, there's no doubt that the vaccine prevents transmission. The data is just so clear.
Yeah.
It's a matter of how good it is at preventing transmission, and that might change as different variants come along. But categorically, the vaccine prevents transmission. We've seen numbers drop, and around the world, we've seen numbers drop. It's a matter of like how frequently you need to vaccinate to keep those numbers down.
We've got a great anonymous question, and it relates to a comment you made, Mark. I think when we asked a question about vaccine efficiency with Omicron, you made some predictions based on our experience with Delta, and that was that although the new variant the vaccine may have reduced efficacy in preventing transmission of the variant, it should still be effective for severe disease and hospitalisation. So could you explain, why you make that prediction based on Delta, given we don't have yet a whole amount of data on Omicron itself?
Yeah, to be clear, yes, it is a prediction or speculation. So the reason being is that the threshold to prevent you getting infected, which blocks you from transmitting the virus, is much higher. So you have to have much higher antibody levels, a much better immune response that will prevent you from getting infected, making virus, and then transmitting it. The degree that you need to, I suppose, constrain the virus to stop it causing the disease is lower. So you can still get infected, but that immune response will contain it to a degree where, okay, you'll transmit it, but you won't get severely sick because your body has got a head start and will be able to fight. So again, that's speculation based on Delta, where we know that's exactly what happened. The vaccine was still very effective in preventing severe disease and death, but didn't block transmission to quite the degree it did with Alpha.
And I heard some WHO commentary this morning that it's likely to be days rather than weeks or months until we begin to get that data on Omicron. So we should be able to get a sense of how transmissible it is, how severe the disease is, and how effective the vaccine is over the next days and couple of weeks, which is exciting. It's amazing how quickly science moves in 2021. Another question from Kerry. Is there any research being done as to what is causing the adverse events post-vaccination, particularly the events that have unresolved symptoms post 120 days after a first dose of AZ? So are there lingering side effects that we have to be worried about? Mark?
For the most part, all vaccines have got side effects, and we've never had a vaccine that's gone out on this global scale so quickly to so many people. So we always anticipated a degree of side effects, but they're very rare. In fact, those side effects that we see with the vaccine are mirrored by the virus itself, except the virus is much more likely to cause those severe side effects. So if we're talking about the rare clotting disorders that we see with AstraZeneca, the likelihood you have clotting disorders with the virus if you get infected are 100 times higher. In terms of the inflammation of the heart that we occasionally see with the RNA vaccines, the likelihood that you get that if you're infected by the virus are again 100 times higher. The likelihood that you'll have ongoing sort of lingering illness and symptoms if you get infected with the virus are obviously way higher than if you had the vaccination. So why does this happen with vaccinations? Very hard to predict, very hard to understand, but they're rare. That's the most important aspect of it. They're rare.
So and I know this is true of the way a lot of scientists think about probabilities. I think for the vast majority of our staff, I think we are up to 98 or 99% vaccinated. We've made the decision that much better to risk an extremely rare side effect from vaccination than a much more likely illness or death from COVID. That's the kind of equation we make as medical researchers when we think about vaccination.
Yeah, and as Vanessa said, it's completely unpredictable, those that get severe disease from COVID, unpredictable. So we're seeing very young people that never had massive viral loads, still having lingering symptoms for a long time.
Yeah, and it doesn't really, I've heard that a lot too, that people who thought that they were very fit might be in the kind of prime of their life, like me at 57, the prime, are getting ill. Never thought it would happen to them. People in their 30s who go to gym every day, really being struck down. So, you know, being fit and in the prime of your life doesn't protect you. All right, a really good question about all of this wastewater stuff. Does COVID really survive all the chemical treatment in waste plants, or are the tests done pre-treatment? What are they actually looking for, Ivo? Are they looking for live virus in our treated water that is capable of infecting, or are they looking for bits of leftover virus?
So
what's it all about?
What they're looking in wastewater testing is really for fragments of the viral DNA, the genetic material of the virus, and the genetic material is quite stable and therefore does actually maintain itself in the wastewater. That can be sampled from wastewater. They are actually have concentrated quite a large amount of wastewater to get one sample for testing, and they have a very ingenious device that they are using, which they call I can't say it. Not a torpedo, but something with poo. I can't remember the name. It looks like a torpedo that they are dragging through the sewage, and that then concentrates the viral DNA that they can collect. And so that's what we're looking for fragments of the virus.
I vote Ivo Mueller is dragging the poo collector around Werribee treatment plant. I think that is a great thing for senior scientists to do. Get down and dirty with the science. We have got another great question from an anonymous attendee. In the survey, when you say recovered from COVID, do you mean an actual 100% recovery, or are they still somewhat suffering from mild symptoms like loss of taste and smell? So I guess a question maybe for Vanessa or for Mark, what does recovery mean? You know, are there people that kind of get their health back but have some of the symptoms longer term from COVID?
Yeah, I think we're still figuring that out. We tend to use that word colloquially in our study. We really mean previously infected individuals or convalescent individuals, but just perhaps speak more colloquially to say COVID recovered, in that they've recovered from infectious disease. But as is very clear, not all of them have recovered. And indeed, you know, in those early days when people were coming in the first month or two, they were doing okay. And then as they were really returning to work after, you know, lockdown ended in Victoria, that's when we really started to see the struggle in people. So yes, unfortunately, a small proportion of volunteers in our study are not recovered from COVID and dealing with the daily impact of long COVID. Yeah.
I am going to go with the last three questions. One for Mark from David A. Given the large proportion of Victorian clusters that are related to schools at the moment, and I guess that's because kids are a less vaccinated population, especially primary school kids that are not yet eligible for vaccination. Given that, do you think that the protocols used to respond to a case at schools need tightening up? E.g., the definition of close contacts, minimum time required to isolate, timing of negative PCR tests. From a layperson's perspective, it seems the current protocols are insufficient. What do you reckon, Mark?
I'll give part of the answer and then I'll throw to Ivo. So Ivo and the others did the modelling around vaccinating children, and he can explain that. From my perspective, kids for the most part aren't going to be super spreaders. It's young adults who will be the super spreaders. They're the ones that go to nightclubs. They're the ones that visit many, many different families. In terms of the schools, because kids aren't a massive source of virus, they don't have this huge amount of virus in their noses and their throats. They're only sort of their closer contacts are going to be problematic. But DHHS has had tremendous experience. They know how many kids have infected other kids, and it's not astronomical. So the initial response was to close down schools. And based on a lot of new data, they're not doing that anymore, primarily because there wasn't merit in doing that. So they weren't seeing massive clusters at schools. They were containable clusters at schools and generally related to very close contact. So that's how the system has adapted. Ivo.
Yes, so there is some very nice work that was done on what to best do around schools by Nick Scott at the Burnett Institute as part of the national modelling, and that's actually available publicly on the website from the Doherty Institute. And there's really two things we're looking at. On one hand is to have as few kids as possible infected by other kids, and also as few days of school missed by children, because we don't want children to miss school. And what Nick has shown very well is that a strategy where we don't close schools, but test children that are contacts and let them come to school as long as they're testing negative, is probably the best compromise between having the fewest days of schooling lost and having only a very small increase in the number of cases compared to closing school and isolating contacts at home for 14 days. And that is the rationale behind why some of these procedures at the HSS and elsewhere are changing now towards keeping schools open rather than closing them, to strike that balance between the loss of schooling versus the number of cases.
Great answer. Two more questions I am going to ask. I think this is the two questions. Great question from Dr. McCullough. Would it be fair to say that our response to COVID is more about a reflection of our society rather than science or the virus itself? How much has science really impacted our response to the virus? Or is it just that in Victoria we are a group of rather obedient citizens? Come on, Mark, give me an idea where science has come in. I thought it was pretty remarkable that we went from a virus to a genome within a month, a genome sequence, and then from a virus to I think the first vaccine was tested 100 days after the virus was discovered. Is that right?
Pretty so. And the answer to this question is complex, but it's got to do with the fact that everything was reacting to something. So you react to a number that comes up, 100 infections in a day. You react to that, and the reaction sometimes is driven by an element of science, sometimes driven by an aspect of urgency. So you're going to overwhelm the hospital system very, very quickly. You're going to put nurses and doctors under a massive amount of stress if you don't shut down aspects of transmitting the virus. But I would say that nearly everything that we've done is based on some aspect of science. Obviously, as Doug alluded to, I thought it was amazing that we got a vaccine within 2 years. If you asked me at the start of this pandemic, would we have a vaccine within 2 years, I would have said, no way. No way could we
one vaccine.
Sorry?
Not just one vaccine.
Exactly. So I keep saying I think that was a tremendous human feat, tremendous. And that was a collaboration between many industries, many academics to try and get that out quickly. But in relation to what Victoria's response has been, I think that it was reasonable to have a COVID-0 response very early, and it gave us a lot of time to understand how we were going to get vaccine rolled out, gave us plenty of time to reduce deaths and impact on our healthcare system whilst we were waiting for the most effective vaccine. So I would say lots of science and a touch of reaction.
Very good. And I'm going to make this the last question. How does WEHI work with researchers around the world? Is it a competition or a collaboration between countries and research centres? Vanessa, you've got this huge study on COVID, long COVID and immune responses. Is it the Vanessa Bryant show, or do you work with other people?
Yeah, absolutely. Well, I could say my previous life pre-COVID is working in the space of rare diseases. So I'm all about collaboration globally. And I think, as Mark says, you know, the rapid movement of science really has been about transparency, sharing resources, sharing data. And you know, one of the reasons we were able to get a vaccine so quickly is that most of them didn't start from scratch. There was previous research that they were built on. Is it competition or collaboration? I'm going to say it is both, and both are really important. There's a lot of collaboration, which I love to see. It's how I love to work. Literally people handing over, sharing reagents, sharing samples. You know, we share our samples with other studies because we realize we are stronger together, and each study is slightly different, asking slightly different questions. But the competition is really important as well. You know, by having different groups around the world taking different approaches, using a different approach to science and different reagents, and getting at the same answer, that's how we're fast tracking to a lot of the truth of what's going on. So it really is a combination of both, but I would say the collaboration far outweighs the competition, which makes me very, very happy as a researcher, because that's how I love to work.
It is far more fun to work in collaborations.
Absolutely.
And I think that's something that WEHI has tried to embrace in every aspect of its work. And I guess that really is the feeling and the inspiration behind our tagline, which is Brighter Together. And I think we all feel very much that as scientists, we are brighter when we work together and when we're challenged by our colleagues, whether they're here in the Institute or at other places. And I think also the future's brighter together when we work with the community on these big problems like COVID. So on behalf of everybody who attended this panel discussion, thank you to our wonderful panelists, Mark Pellegrini, Ivo Mueller and Vanessa Bryant. Thank you to Casey, Deb and the team that put together this forum. They did a fantastic job. And most of all, thank you to all of you, the audience, who have been really active participants in what I found to be an absolutely informative Q&A discussion. So thank you, everybody. Please stay safe. Enjoy the run-up to the holiday season and vacations. Fingers crossed Omicron turns out to be a variant that is much more mild and that we can all enjoy our hard-earned freedoms in 2022. So thank you, everybody.